Dr. Hanahan, welcome. And it is my honor to introduce you. So, good morning, everyone, and happy 2026. Welcome to the Myeloid Network series. My name is Dr. Jelani Zarif, and it is my true pleasure and honor to introduce today's speaker, Dr. Douglas Hanahan, distinguished scholar of the Ludwig Institute for Cancer Research, working in its Lausanne branch in Switzerland. Dr. Hanahan is an emeritus professor of molecular oncology and former director of the Swiss Institute for Experimental Cancer Research at the Swiss Federal Institute of Technology in Lausanne. In addition, he is the founding co-director of the Swiss Cancer Center in Le Mans and played a leading role in shaping the Agora Translational Cancer Research Center there, where he now directs a translational cancer research laboratory. Dr. Hanahan received his bachelor's degree in physics from MIT and his PhD in biophysics from Harvard University, where he was supported by the Harvard Society of Fellows. He conducted early work at Cold Spring Harbor Laboratory, both as a graduate student and a faculty member, and later spent more than 2 decades at UCSF before joining EPFL in 2009. Throughout his career, Dr. Hanahan has made several landmark contributions to cancer biology. He developed methods that advanced molecular cloning in E. coli. He pioneered genetic mouse models, and together with Judah Folkman, discovered the angiogenic switch, which enables tumors to grow and progress throughout malignancy. In collaboration with Bob Weinberg, he also introduced the influential framework that I've required most of my undergrad students to read and graduate students to read in my laboratory, The Hallmarks of Cancer. And they've done an incredible job keeping that updated throughout the years. And it continues to guide our field today. So today, Dr. Hanahan will talk to us about disrupting IL-1 driven systemic immunosuppression by neutrophils, enabling vaccine efficacy in HPV-16 tumors. Please join me in welcoming Dr. Doug Hanahan.
Okay, thank you for that lovely introduction. Yeah, so I'm going to, given the theme of this series and focus on myeloid cells, I want to describe a new study on myeloid cells, neutrophils in particular, that's come out of my lab. And it's a long saga, as I'll mention. So let's see here. Okay, so basically, we've been modeling in mice, as I'll describe, cervical cancer. And it's a logical target for therapeutic vaccines because this is a DNA tumor virus. It infects the cervical and epithelia and other squamous epithelia, goes through an episomal form, but then integrates a fragment of the genome, including the oncogenes, which drive a proliferative response that can result in progression of intraepithelial neoplasia and invasive cancer. And so basically, this disease is driven by a virally encoded oncogenes, oncoproteins E6 and E7, which abrogate PRB and P53 to drive tumorigenesis. They're immunogenic neoself-antigens, so it seems that they're logical targets for vaccines. And in fact, the decades of work have gone into trying to develop vaccines for cervical and other HPV driven cancers. The problem is that they don't work. So this is just one clinical trial here in advanced recurrent HPV-16 positive cervical cancer patients, comparing the vaccinated group and the matched cohort. And so there's really, and this is a relatively recent vaccine, but there's a whole series of them that have been tried and failed. So something is going on. And so this is which is limiting the ability of these vaccines to have efficacy. And so we came into this because my lab has been modeling, as was mentioned, cancer in genetically engineered mice. And so one of these is the so-called K14 HPV-16 mouse model of cervical and skin cancer. So in this model, we took a keratin-14 gene regulatory region and aligned it to drive the early region of HPV-16, including the E6 and E7 oncoproteins. And basically, this was work that was done at UCSF. Jeff Arbeit and Lisa Cousins were the key principals in this. And essentially, making a long story short, male and female mice develop skin cancer, which goes through periods of hyperplasia and dysplasia. And female mice, who's in the context of sustained estrogen levels, develop cervical intraepithelial neoplasias, which progress into cervical cancers. And so that's just and so then these cervical neoplasias and cancers are histologically very similar to those that are seen in the human cervix. So actually, amazingly enough, I mean, I've been working on papillomavirus transgenic mice for 40 years. First made a mouse model in 1986 at Cold Spring Harbor involving the bovine papillomavirus oncogenes. And then in the 90s at UCSF, we developed this model of cervical and skin cancer. And so we studied these in the 90s, and then there was sort of a hiatus for 20 years, and then it was sort of, and then we renewed the focus on these in this decade based on the development of new knowledge. So again, just to reiterate, I got interested in this when I was at UCSF. That again, with the rise of checkpoint immunotherapy, that I thought that HPV-16 expressing tumors should be a poster child for cancer immunotherapy. This was even before the trials I just mentioned had failed. And again, same reasoning. The oncogenes drive tumorigenesis, and the oncoproteins are immunogenic neoself antigens. And so so I really felt that this should be a platform for for testing immunotherapies. And in fact, in the mid 90s, I went over to would drive across the bridge to Berkeley to where Jim Allison's lab was, and get aliquots of CTLA4, his prototypical checkpoint inhibitor, take them back to UCSF and treat these mice, and there was no efficacy. And so that was rather disappointing. Meanwhile, of course, he went on to show in transplant models that CTLA4 had benefit, and the rest is history. But I never sort of gave up. And for the students in the audience, so this failure of why did it succeed in these transplant models and why did it fail in this gem led us to sort of reinitiate an investigation of this after a long hiatus during which we moved to Lausanne. And because basically what we found above and beyond this early study with CTLA4 is that we started working in Lausanne with colleagues in the bioengineering department who were developing vaccine technologies. And we studied a vaccine based on the E7 oncoprotein. And essentially, um, this is a nanoparticle-based E7 long peptide vaccine. It's very effective in a tumor transplant model called TC1. And so here you can see this tumor is full of CD8 T cells. And so this is very similar to the responses that Jim Allison was seeing in other tumor transplant models with checkpoint inhibitors. But basically, this transplant model was very sensitive to this vaccine. But when we took the same vaccine, which was producing this dramatic effect, and looked and treated the mice with de novo cervical carcinomas, there was basically no effect. I mean, so here the red here is showing nests of cervical cancer cells in the cervix, and the green are CD8 T cells. So there's really no action. And again, this I believe is replicating the poor responses to E7 peptide vaccines in advanced cervical cancer patients. So this dichotomy really led us to, having shown that this vaccine really worked well in a transplant model, which we published in 2018. Then we went on to show in 2020 that even though it worked in the transplant model, it didn't work in the GEM. And so the question was, well, what's going on there? And so what I'm going to describe now is the line of investigations which led us to some understanding of what's going on. And in essence, what we think is beyond the well-established immunosuppression that one sees in the tumor microenvironment, these tumors are developing systemic immunosuppression, which affecting all of the lymphoid organs. And so the systemic immunosuppression appears in the early stages of HPV-16-induced carcinogenesis in the skin and in the cervix, comes on quite early, long before there's a tumor microenvironment that's immunosuppressive. So these are hyperplastic and dysplastic lesions in the skin, so SYN1 and 2 lesions in the cervix. So it comes on very early, which led us to really suspect that this had something to do with the expression of the viral oncogenes. And essentially, the systemic immunosuppression is manifested by a big expansion of CD11 B cells in spleen and lymph nodes and in bone marrow, and they're principally neutrophils. So, or myeloid cells in your metric. So, and I'm not going to show you the data, but these cells are immunosuppressive in a CD8 co-culture assay. So this was the initial data uh, that really led us to think, well, we need to focus on these immunosuppressive neutrophils that are in all of the lymphoid organs, in addition to being in the tumor. So the question is, what are the underlying mechanisms of SIS, and can we disrupt it and enable therapeutic vaccine responses? And so basically, at this point, a PhD student, Morgan LeContre, and a postdoc, Jeremy Gillot, took on this project to try to figure out what was going on. So they worked together as a very effective team the last 5 years, really trying to figure out what's going on. And so the hypothesis was that the neoplasias, as well as tumors, were secreting ligands into the circulation, which were basically binding to receptors in the bone marrow and spleen and eliciting the expansion of these immunosuppressive neutrophils. So they performed a series of multi-omic screens. They looked at the secretome of secreted proteins that were released into the blood using proteomic and analyses, cytokine arrays, 10x single cell sequencing in the cervix to look for secreted ligands, and then looked in the bone marrow and spleen again by single cell sequencing for receptors for those ligands. And so the data that came out was interrogated in several instructive tumor-derived cell lines, where we filtered these ligands. And so basically, what they developed was syngeneic models that had much different immunological phenotypes. And the key ones are called IMP for immunopermissive and IS1 for immunosuppressive. And so the IMP line does not develop SIS. It has an abundance of macrophages and very few neutrophils. The IS1 line develops systemic immunosuppression and has an abundance of neutrophils. And so in this multi-omic screen, there were 93 candidate ligands that came up, but they segregated differentially into the immunosuppressive line, the immunopermissive line, and then actually there was an intermediate line here, which has sort of a mixture of both. And so the question then was, well, how to filter this down? And so what Morgan and Jeremy did was to basically cross-filter the 44 ligands that were expressed in this immunosuppressive cell line and ask, well, which of these ligands had receptors on neutrophils? And that matrix produced 27 ligand receptor pairs, of which there are just a variety of them shown here. But to make a long story short, I'm just going to focus on the IL-1 family because we decided for a number of reasons to focus on the IL-1 family, and in particular, three members of this cytokine superfamily called IL-1 alpha, IL-33, and IL-36B. And so the question was, well, are these ligands involved in eliciting systemic immunosuppression? Which is basically the question asked here. So I'll show you several pieces of data that argue that they are. And so one of those, we took normal mice with no transgene, no neoplasia, and inoculated them with a cocktail of these three IL-1 ligands. We've also done things with individual ligands, but the key is that all three of these ligands are contributing. And if you commit, so we combine them into a cocktail and inoculated normal mice, did FACS analysis, and did CD8 co-culture assays with splenic CD11b neutrophils. And so basically, what we found was that these three ligands could elicit an expansion of neutrophils. And so that's shown here, that basically the IL-1 combo elicits an expansion of neutrophils in the spleen. And it's similar to what happens in the de novo tumor-bearing HPV-16 mice. So again, which again elicit this, as I already showed you, this expansion of neutrophils in the spleen, bone marrow, lymph nodes, etc. GCSF, which is a logical suspect, had only a modest effect. And then if you look at proliferation in co-culture, well, first of all, splenic neutrophils from control H2B mice did not inhibit CD8 T cell proliferation, but mice, normal mice that were treated with this cocktail were immunosuppressive, or suppressive of T, or specifically suppressive of CD8 T cell proliferation in co-culture. Now, as I mentioned, this immunopermissive line is not immunosuppressive, does not produce splenic neutrophils that are immunosuppressive. However, if you treat them with this IL-1 cocktail, now they become potently immunosuppressive. So again, these are tumor-bearing mice from the immunopermissive line. They're not no SIS, no inhibition of proliferation. You treat them with the cocktail of these ligands, they're potently immunosuppressive to a similar degree as the immunosuppressive cell line. So this was really suggesting that these three ligands are sufficient to elicit this state of systemic immunosuppression. So, and there was other evidence for this, which is actually implicit in what I just showed you. But I just wanted to walk you through that. If you look at the immunopermissive model, as tumors grow, um, and you look at CD8 T cell proliferation and from splenic neutrophils, and you look at the abundance of splenic neutrophils, that basically there's no major effect on CD8 proliferation in neutrophils that are derived from tumors that differ in growth states, and there's no big expansion of neutrophils. And so this is again there. So there's really no systemic immunosuppression in this context. But if you overexpress in this immunopermissive cell line this triplet of IL-1 ligands, then you see a much different effect. As the tumors grow and they reach a certain volume, they become the splenic neutrophils become potently immunosuppressive for T cell proliferation, and there is a big expansion of neutrophils. So again, this line switches from this phenotype characteristics to one of immunosuppression with an abundance of neutrophils. And this is quite similar to what we see in the IS-1 model, where again, as the tumors grow, they develop the ability to inhibit CD8 T cells, and the neutrophils expand. So basically, it appears that from both of these lines of investigation, that these three ligands are capable of eliciting this state of systemic immunosuppression. Okay, so then is this actionable? Well, it's notable that this IL-1 superfamily talks to a series of different receptors, and in particular, IL-1 alpha and IL-1 beta talk to one receptor, IL-33 talks to another, and IL-36 beta, along with some of its family members, talk to a third one. But what they have in common is a signaling receptor called IL-1 RAP. And so then this signaling receptor is necessary for downstream signaling. And so you have a ligand binding domains that are specific for the different ligands, and then you have a common signaling receptor, IL-1 RAP. And so this suggested the potential for simultaneous blockade of all three ligand-induced signaling pathways. So where is IL-1 expressed? Well, it's primarily expressed in neutrophils. So again, doing single cell analysis, I mean, it's mainly expressed in the neutrophils, in the bone marrow, in the blood, in the spleen, and in tumors. So it's highly specific and therefore seems a reasonable target to consider in line of this mechanism. So basically, we acquired from a biotech company in Lund, Sweden called Cantargia, an anti-IL-1 RAP monoclonal antibody. And so if you treat, for example, tumor-bearing mice derived from the IS-1 cell line, which develop SIS, and you treat them with anti-IL-1 RAP, you modestly delay tumor growth. It's not dramatic, but you see a modest delay. But there's evidence again for target modulation in the sense that there's a reduction in CD11B cells, there's a reduction in neutrophils, and there's a reduction in the neutrophil to T cell ratio. Again, which is consistent with fewer immunosuppressive neutrophils and therefore more CD8 T cells. So this is encouraging, but obviously as monotherapy, not dramatic. But it's suggestive that in fact this is having an on-target effect of hitting these immunosuppressive neutrophils. But the exciting result then was that if we combined anti-IL-1 RAP with this nanoparticle E7 long peptide vaccine, now we get combinatorial benefit. In this particular model, there's a very modest effect of either of the monotherapies, but we get a stabilization of disease with the combination. So this is really unlocking the capability of the vaccine. And this can also be seen in a survival trial. Basically, anti-IL-1 RAP and by itself has essentially a minimal survival benefit. If you include an isotype control with the peptide vaccine, again, no significant survival benefit. But basically, there is a significant, but not very. It's statistically significant, but not very profound benefit. But if the combination then produces really a marked survival benefit, and this is all dependent on CD8 T cells. So again, this is the response you get with a double combination. And then if you layer on anti-CD8, therapeutic efficacy is obviated. So it's clear then that the combination of IL-1 RAP, which is abrogating the systemic immunosuppression, along with the vaccine, is eliciting a T cell response that is responsible for this improved survival. And in terms of molecular effects, I mean, if you look in the tumors, you see an increase in CD8 T cells, an increase in effector CD8 T cells, and actually we can look at E7 specific CD8 T cells using tetramers. And so again, in all cases, the anti-IL-1 RAP plus the vaccine is increasing the overall number of CD8 T cells, the number of effector T cells, and antigen-specific CD8 T cells. Again, consistent with this result here that CD8 T cells are responsible for the therapeutic efficacy. And interestingly, now IL-1 alpha and beta have been really coming on the radar from many labs now as having effects on tumors. And there have been a number of studies suggesting that IL-1 alpha and or IL-1 beta are the instigators of basically immunosuppression in tumors. There's been very little work done on systemic immunosuppression. We're sort of pioneering that, but others have shown that IL-1 alpha or beta is also instructing in the immunosuppressive tumor microenvironment. And indeed, we think this triplet combination is also instructing an immunosuppressive environment in the skin and cervical tumors that we're studying. I'm just not taking you through that data for lack of time. But the key point here is, in contrast to some of these other studies, is that if you inhibit IL-1 beta or IL-1 receptor, which is what is important for IL-1 alpha and beta, they don't produce the same effect. So you can see here that IL-1 beta or the IL-1 receptor is not as efficacious as anti-IL-1 RAP. And so this leads us to conclude that in these models that the triplet combination is actually important. We haven't fully illuminated what the different components of it are doing, but it does seem that that IL-1 alpha or IL-1 beta are not sufficient. Now we went on to show that deja vu to the 1990s that at UCSF that actually anti-CTLA-4 actually does have benefit if you add it to this triple combination. And so essentially here again, this is what I showed you previously. And here's the doublet of anti-IL-1 RAP and the vaccine. And if you layer on CTLA-4, you get a substantial increase in survival. The monotherapy is not shown here, but CTLA-4 still doesn't do anything by itself. So that's, uh, but anyway, it's come full circle that CTLA-4 does have value. And this, of course, is rather satisfying for me because I was so convinced in 1994 that CTLA-4 was going to have an effect in these models, and it did not. But it took us 30 years to understand why. But anyway, we didn't give up. We found an explanation, and so that is satisfying. This sort of just to say that if your experiments don't work and you think they should, maybe if you're persistent, you'll find a solution. Okay, and so the other thing that we did was bioinformatically was develop a small gene signature of this IL-1 induced systemic immunosuppression. And so it involves 10, and so again, this involved filtering, genes from comparing the IS-1 and the IMP cell lines, and they were differentially expressed along with the 93 candidate ligands. We came up with a 10 gene signature, which includes the 3 IL-1s that I just described, along with GCSF. We do think it's involved. It's just not the driving force, along with several other well-known instigators. And so this 10 gene signature has some prognostic significance for human cervical cancer. So if you go into the TCGA database for cervical cancer, if you have a um, uh, this is actually, I think, backwards. Now that this is we had some problem with the graphics here, but anyway, the this is the patients that have a high SIS signature, and and these are the ones that have a low one, and the SIS signature score, really tracks with the number of neutrophils. So again, it argues that that this SIS signature is prognostically significant in human cancer, and then therefore that systemic immunosuppression is operative in some subset of human cervical cancers. And it's also true in other squamous cell cancers. Again, I think this is the color code is backwards, but the red is a high signature score, and this is head and neck cancer, lung, squamous cell cancer, and bladder cancer. So in all cases, there's indications that a subset of patients with these tumors have a high SIS signature, which we associate with increased neutrophils and implicit systemic immunosuppression in these individuals. And so the question now is, like, if we look in cervical cancers, you can see some tumors have lots of CD8 T cells and few neutrophils. Others have lots of neutrophils and few CD8 T cells. And so we're thinking that we could basically do screening for patient selection and in terms of thinking about using vaccines, because we would predict that patients that have a low SIS signature are going to have an abundance of T cells and are going to be more responsive to vaccines than to ones which have a high signature. So that's one of the things we're thinking for the future is how can we translate this? Okay, so basically, uh, to conclude, just to summarize what I've told you, and then I want to have a few translational comments, but that blocking IL-1 alpha, IL-33, and IL-36 beta using anti-IL-1 RAP impairs tumor growth and decreases immunosuppressive neutrophils. It enables an E7 long peptide vaccine to be responsive in this immunosuppressive tumor-bearing mice, concomitant with the induction of tumor-specific CD8 T cells, which are necessary for the impaired tumor growth and survival benefit that we see. We've derived an SIS signature for the systemic immunosuppression, and it is associated with worse prognosis in advanced cervical cancer patients. And notably, cervical cancer is the fourth leading cause of cancer deaths in women worldwide. So this is a global health problem, but it's not only in sub-Saharan Africa, it's in much of the second world, South America, India, China, throughout the second world. Cervical cancer is a major leading cause of cancer deaths, and there are no good therapies. And so, we're thinking that a triplet therapy of an HPV-16 oncoprotein vaccine, anti-IL-1 RAP, or another pan-IL-1 inhibitor and anti-CTLA-4 warrant consideration for clinical trials. And we're certainly hoping to try to instigate that. Now, just to further elaborate on that translational rationale, as I mentioned, we got this antibody from Cantargia, this biotech company in Lund, and they have a clinical version of this called nadunolimab, which is already in phase 2 clinical trials. And so it's safe and it's showing efficacy. It's not being used at this point to treat systemic immunosuppression because this is a new concept, but it's being used to target essentially the immunosuppressive tumor microenvironment in PDAC and non-small cell cancer, lung cancer patients. But the key thing is some of the data from those trials, again, that if you have patients, which have back to this idea here, that if you have patients that have a lot of neutrophils, how will they respond to IL-1 RAP? And basically, so patients that have a high neutrophil count, such as the one I just showed you, that in that case in tumors, on treatment with IL-1 RAP, there's a drop in the neutrophils after some period of time, and the number of lymphocytes begins increasing. And then again, the neutrophil to lymphocyte ratio is going down again, which is sort of a prognostic factor for non-responsiveness. So this is encouraging that IL-1 RAP has the ability to abrogate SIS in patients. Now, interestingly, as I mentioned, there continues to be clinical trials with new and improved HPV vaccines. And there was one recently done, I don't think it's published yet, from Keith Malif and his colleagues. But what they found was that again, high blood NLR defines HPV-16 patients who do not respond to therapeutic vaccination, in this case combined with anti-PD-1. So consistent with the data I just showed you and indicated that if you have a high NLR ratio, that suggests SIS, and indeed those patients are not responding. So I think these results support a rationale for testing our clinical combination therapy and our triple combination therapy in future clinical trials, and maybe involving patient selection with this SIS signature to identify the patients that could most benefit from this triple therapy because they are suffering from systemic immunosuppression. So this study was published in March of this year in Cancer Discovery. And as I mentioned, Morgan LeConte and Jeremy Gillot were the key players in this, along with Rochelle Marconi, a bioinformatician, and our colleagues at Cantargia. So I'm at that point, I'm happy to take questions. And I also think that Morgan and Jeremy are online, and they can also answer questions potentially. So thank you.
Thank you, Dr. Hanahan. Outstanding talk. We have, and for those who are listening in, please, if you want to raise your Zoom hand and ask the question, you can. We have a few in the chat. So Maria Caffarel, would you like to answer your question?
Yes, thank you. Hi, everyone. Can you hear me?
Yeah.
Yes.
Many thanks, Doug, for this excellent talk. I was wondering if IL-1 beta is part of this combo inducing systemic immune suppression. Because you only mentioned IL-1 alpha, but I would like to know your
Yes. So in
results.
fact, IL-1 beta is induced as well. So it's actually there's actually all four of them are involved. The IL-1 alpha did not. IL-1 beta seems to be produced in different cells. That it's not produced by the cancer cells, but it is present in the circulation and in the tumor microenvironment. So yeah, we do think that IL-1 beta is part of the mix.
Thank you.
Judy?
Hi. Uh, Great talk. I have a couple of questions. Do you know what the neutrophils are doing to promote tumor progression besides preventing response to vaccine? Have you specifically identified the mechanisms?
I don't think so. Jeremy, Morgan, do you have anything to add to that?
Hi. So what we mostly characterized was the suppressive effect on the T cells. So what we can say is that the neutrophils are basically blocking T cell activity as well. So the induction in the periphery, but also their activity in the tumor microenvironment. So this is what we think is the main mechanism.
I'm just curious to know if you know the molecules involved in that.
I mean, we
Yeah,
haven't.
so is it specific cytokines? Is it yeah.
Yeah, so some previous lab members tried to block, for example, arginase and some specific molecules that are released by the T cells, but none of those could fully restore T cell activity. So it's probably a combination of factors.
Okay, thank you. I had another question about the IL-33. Did you try to block IL-33 directly and know whether that has an important role?
So we haven't done that yet, but this is something that we are planning right now. So yeah,
Okay,
this is
great.
the type of experiment we're going to do.
Terrific.
Yeah, because in
Thank
fact,
you.
IL-33 can have beneficial effects on T cells as well. So it's a
Yeah.
complicated, sort of context dependent. Another sort of embellishment to your point is actually decades ago we showed that neutrophils were also pro-angiogenic, and we haven't really delved into the
Wow.
possibility that because what I didn't show, of course, was that we've identified, or Morgan and Jeremy in particular have identified sort of different classes of neutrophils in the tumors, and including sort of these N5 and N6 neutrophils that Pate has described. But I think it's an interesting question for the future of whether they have other tumor promoting activities beyond immunosuppression, but we have not had the ability to really answer that.
Thanks.
I think Alexandra has a question.
Hi, great talk. Thank you so much. Just a question about, have you ever tried to block neutrophils directly or some of the neutrophil products that cause inflammation like S100, A8, A9?
We did in the 2020 paper. We went through a whole catalog of things, but none of them seemed to work. But I
Yeah.
don't know. I'm not sure we've done anything more recently. Morgan, unless you could correct me if I'm wrong.
So S189, we haven't done anything, but what we tried was to deplete neutrophils using LY6G depleting antibodies. And this has a very, very transient effect in our model. So after a few days, there is a really harsh rebound of neutrophils. So we were not able to deplete these cells long enough to actually see, like an effect long term, let's say. And we also tried to block GCSF, and blocking GCSF on its own also induce rapid resistance mechanism or relapse in terms of rebounds in neutrophil counts. Yeah.
Yeah,
Okay,
we
thank you
tried
so
actually
much.
in the
Yeah.
2020 CIR paper. We also went through, cocktails of LY6G and GR1 and all these things. And again, it was the
Yeah.
same story. They
Yeah.
couldn't really sustainably eliminate the neutrophils. But this, I think, is a common theme in myeloid biology now, that it's very difficult to ablate macrophages and neutrophils. Really, the future, I think, we all probably believe now is reprogramming them rather than
Yeah.
than trying to ablate them.
All
Yeah,
right,
because also,
yeah.
just to add one point, so it was not shown in today's presentation, but actually, so we see a reduction in the numbers of neutrophils in the periphery, but what we really see and is more striking is the fact that those neutrophils are reprogrammed and they are not immunosuppressive anymore. So we think that our strategy is different because it reprograms the neutrophil rather than depleting them. So
Thanks so much. I asked just because I'm going through the same problem now, so good to hear everybody does the same thing.
Okay, Jelani, do you have a question?
Yes, fantastic talk. I was curious about netosis and any other processes of these neutrophils. Have you all looked at that? That could be a way that changes the neutrophils after treatments.
we have not looked into netosis in this published study. So maybe in the future it would be interesting to look at it as well. But at the moment we don't have any data supporting activity on netosis.
Thank you.
Dirakin, do you want to turn on the camera and ask your question?
Yeah, very nice talk. So I have a question. So because you said these neutrophils they are in the systems can affect the CD8 functions, and you also show the neutrophils in the tumors. So I'm thinking about I'm wondering what is necessary the neutrophils need to be in the tumors to block CD8 function, or it's not necessary the tumor recruit the neutrophils in the tumor?
So it's a very interesting question. The problem is we don't have a model in which we have systemic immunosuppression and no neutrophils in the tumor. So usually it goes hand in hand. If we have neutrophils in the tumor, it doesn't mean we have systemic expansion. But if we have systemic expansion, usually we tend to have neutrophils in the tumor microenvironment. So it's difficult to answer your question because, yeah, for that reason.
Yeah, as I mentioned, we suspect we're quite, again, also from the profiling that they've done, we think that there are definitely tumor promoting immunosuppressive subtypes of substates of neutrophils in the tumors, but we have no way to separate them from the systemic ones.
Yeah. So in your combo treatment, did you see like the CD8 more recruit in the tumor, or just like because you still need the anti-CTLA-4 to expand your efficiency? So I'm curious how the CD8 in the tumors after your combo treatment, are you see more CD8 in the tumor?
Yes, indeed. We do see an increased influx of CD8 T cells in the tumor that are, tumor specific, so E7 reactive. And in the triple combo, we have an even greater influx compared to the double combo.
Okay, yeah. Because it's interesting because I saw the treatment they just expand the lifespan, but not real to rescue the tumor. I'm not doing the tumors. I'm curious why all the treatment can rescue the mice, but they just expand the lifespan. I'm just curious.
Yeah.
It's an excellent talk, and I want to just ask a follow-up question on this. So I know it's more evaluated in spleen and lymph nodes, but do you see any changes in macrophages and other immune components after this anti-IL-1 RAP treatment? The NK cells, they are also immunosuppressed by neutrophils or just T cells?
This is a very good question. So we haven't looked too much into NK cells. So the only thing we know is that the number of the NK cells doesn't change upon treatment. So the treatment we are applying doesn't have an effect on the NK cells proportions. But yeah, in this study, we really focused on CD8 T cells because we work with the therapeutic vaccine. And so we will look at the response to the vaccine. So for this reason, we look at antigen-specific responses.
Great. So next question from Paloma.
Yes. Hi. Can you hear me well? Because my microphone was not working well before, but
Yeah,
I think now it's good. Okay, so thank you for your talk. It's really nice, and I recently read the paper and really liked it. But I was wondering if you have checked for the effect of IL-1 in metastasis of your models, if this blocking the IL-1 RAP has an effect there, or maybe it's not so relevant for this type of cancer.
Well, the answer is we don't. These models are not. We can't. These aren't good models of metastasis. So unfortunately, it's a great question for the future, but we don't. We haven't done anything.
Thank you.
Paco?
Yeah, thank you. Very nice work. I missed the first few minutes of the talk, so maybe my question might be redundant, but I wanted to ask you first, how is there any evidence that actually this systemic immunosuppression might be protective of tumor initiation in a more broad context?
Well, that's an interesting question. I mean, we think the systemic immunosuppression is elicited by the expression of the HPV-16 oncogenes in keratinocytes. Because again, if you express the HPV-16 oncogenes in other epithelia, for example, of the TC1 cell line, which is a kidney epithelial cell line, it's not immunosuppressive. But essentially, the HPV-16 oncogene expression in keratinocytes, which is their biological target, elicits this SIS. And so we think this is probably the virus has evolved to be able to do this, to sort of limit the immune attack so that it can go through its life cycle. So it's sort of so the question is, so the SIS doesn't come up until you already have an incipient neoplasia. Whether it could be protective is an interesting question, but essentially, qualitatively, what we see is immunosuppression. So
But
there's
I'm
no.
wondering, yeah, I'm wondering whether independently of the tumor initiating events, there might be comorbidities or other conditions that might actually result in a similar systemic immunosuppression that might favor tumor development even when the tumor is initiated with the first mutations.
Oh, so yeah, it's an interesting question for sure. Yeah.
Okay,
Quite
thanks.
possible. Yeah.
Questions?
There's one question in the chat by Mark Exley. If Mark would like to ask the question.
Thank you.
There you go.
It was more of a comment than a question. Just noted that René Brentgen's group had also recently published that IL-36 gamma, in their case, reprograms neutrophils when it's secreted from CAR T cells have been transduced with it as an armoring as well. So it seemed like very consistent because you had IL-36 in there as well, right?
Okay, interesting. Yeah, we got to find that.
Yeah, just out.
Okay, cool.
Enjoy.
Thank you.
Thank you.
I had a quick question. So we've studied HPV expressing head and neck cancer models and did a little analysis of the inflammatory cytokine expression in HPV positive patients and found that you can divide them into those patients that have a high inflammatory signature that live for an extended period, well past 5 years. And then there's a population that do very poorly. That's 65% that would have some immune suppression. Is there any similar dichotomy in cervical cancer patients that you know of that could maybe account for you pointed out there were patients that didn't have a lot of neutrophils, so I just wonder what the statistics might be.
I guess it depends on, this is we all use inflammation, but the question is, are we talking about T cell inflammation or myeloid inflammation? But yeah, certainly the stuff we've done with the signature is suggestive that the patients that are doing better have they may have a greater. We didn't apply a CD8 T cell signature in that analysis, but the prediction is if you did it, that those patients that had a low SIS signature would have had a higher CD8 signature. But it's a good experiment. We haven't done it.
Thanks. And Paloma?
Yes, I just had another question because you mentioned that the anti-IL-1 RAP that you use is probably blocking IL-1 signaling from IL-1 alpha and beta, also IL-33 and IL-36. But how is it different from the IL-1 RAP, like the nadunolimab or the Cano4? Because we also happen to work with the Cano4 ourselves, and what we were told from Kantar here is that it blocks signaling from IL-1 alpha and beta as well as IL-33. So I was just curious about if your anti-IL-1 RAP is structurally different from the Cano4, or how is it different?
Well, I think Morgan could correct me, but I believe the one we're working with is one that recognizes mouse as opposed to the clinical compound. But
Yeah.
as I said, Cantargia, until we came, until Morgan reached out to them and they became aware of it, and we did the study. They, SIS and was not, and even targeting neutrophils, I don't think was high on the radar of their clinical rationale. But I think it's now turning out that, as I showed you with that little bit of data on from their clinical trial, it turns out that it is modulating myeloid cells. But I don't think this was their initial rationale for their studies. Interestingly enough, yeah. So,
thank you.
Any other questions? No? Okay, thank you so much, Jack, for the excellent talk today. And yeah, if you still have any, if anyone has questions, just go ahead and ask. Otherwise, I think we are done with the questions today. So thank you so much.
Yeah.
Okay, great. And thanks to Morgan for her insights and good questions. Thank you all so much.
Thank you all.
Thank you.
Okay, bye-bye.
Bye-bye.
Happy New Year.
Happy New Year.